Hello, everyone. Good morning. Good morning, good morning. Good to see everybody. Yes, type your name into chat for me, as usual. I have new glasses on today because they're computer glasses as opposed to my ordinary glasses for distance, because my eyes have been getting very tired and very bleary working on the computer all day. So now I have a prescription specifically for using the computer. All right, good job. Yeah, if you haven't already, go ahead and type your name in for me. That's the way I take attendance here, so. Okay. So today we're going to be talking about oxygen. We're going to be talking about oxygen and its role in the growth of bacteria and how important it is that we know how that particular species of bacteria relates with oxygen. Number one, because it helps us grow it well in the lab, if we know that. And number two, because it can help us identify that microbe, which is one of the things we do in a micro lab. It can help us identify the genus and the species if we know its relationship to oxygen. Now, before I get started in the lab material, let me ask if anybody has any questions. We have an exam this week, so I know you all have been studying. If anybody has any questions about lecture material, feel free to type into chat what your question is. Remember, if you have a question about something, there's a really good chance that somebody else in class has a question about it too. So don't ever hesitate. All right. So let's talk about oxygen for a minute. Humans need oxygen. Humans have an absolute requirement for oxygen. We'll die without oxygen. About how long can we'll call it an average adult. How long can an average adult go without breathing air? How long? Type it into chat for me if you know. Let's say I won't say me. Let's say an adult person has a cardiac event and falls to the ground in the middle of the supermarket. They're not breathing and their heart has stopped pumping. How long do they have before death? Amy's got a guess. Amy's saying about 5 minutes. Anybody else have any idea? How long can you go without breathing oxygen? If you're an adult person, you're an adult and you're, we'll say average body size, average fitness. How long can you go? How long before you die when you're not breathing? SAP is guessing 10 minutes. Any idea? Any idea, the rest of you? You can't go very long, can you, without breathing? And I know most of us are interested in healthcare and the healthcare field. When a person is having a medical emergency like that, and the emergency medical responders come in, they follow a rule of ABC. ABC. And that stands for airway, breathing, and then cardiovascular. First and most important thing is to get that person breathing, clear their airway, and get them breathing, whether you're breathing for them or they're breathing for themselves. The most important thing in that emergency is to get oxygen into them. Candy's saying about 4 minutes, we start to lose cells. You're right. Yep. Yeah, you're talking about minutes. You have minutes of time left when you stop breathing oxygen. Now it gets a little confusing because it does depend on how old you are and how fit you are. It also depends on what you were doing at the moment that you collapsed, if you were at rest, or if you were very active, or somewhere in the middle. But generally speaking, like Candy said, in about 4 to 6 minutes, cells are going to start dying for the lack of oxygen. And certainly by about 10 minutes, you're gone, you're done. And the reason you're done is because you can't get oxygen. You don't have oxygen. Now, tell me why? Why is oxygen so critical to a human? Why? What is it about oxygen? What is the oxygen doing? What is it that the oxygen molecule does in our cells? Do you know? You've got to have it. If you don't have it, you're going to die within minutes. What is it that oxygen does? Amy's saying it helps fuel the cell to keep the cell alive. Yeah, yes. How? What's its role? What is the role of oxygen in fueling the cell? Austin has it. Austin's got the right answer. You need oxygen to make ATP. If you are an aerobic organism like we are, you have an absolute requirement for oxygen in order to make your ATP. Good job, Saab. We need it to make ATP. ATP is what we call the energy currency in a cell. It's what a cell uses it spends its ATP to perform chemical reactions. It's the energy molecule made inside of your mitochondria. You probably learned that when you were in middle school or high school, that the mitochondria are the powerhouses of the cell. That's where your ATP is generated. And oxygen is required to generate ATP. Oxygen is what we call the final electron acceptor molecule. In the long, long line of chemical reactions that make up cellular respiration and oxidative phosphorylation, which is how we make our ATP, oxygen sits at the end of a structure called the electron transport chain. Oxygen sits at the bottom of that chain, and it takes on the electrons that travel down that chain. And it accepts them, it takes them in. Oxygen becomes reduced, as we say in chemistry. If you don't have oxygen there, at the end of that electron transport chain, the chain will stop. And your cellular respiration process will stop. And you will not make any more ATP. All cells, human cells, bacterial cells, archaeal cells, plant cells, fungal cells, all living cells on our planet use ATP as their energy currency. They use it like money. They spend it like money to get work done. Without ATP, a cell cannot survive. And for humans and other aerobic, oxygen requiring organisms, that's the role it plays. It's the final electron acceptor. It takes on the electrons that were systematically removed from glucose. Your cells burn glucose in order to make ATP. And as they burn the glucose, that's the term we use kind of loosely, as they burn or oxidize the glucose, they take all the electrons out of it. They break all the bonds and they take all the electrons out of it. And those electrons have to go somewhere. They have to land somewhere. They can't just be left loose. And it's oxygen that takes them in the end. Oxygen takes the electrons and some hydrogen and becomes water. So if you don't have oxygen, you can't make ATP. And if you don't have ATP, you can't survive. So that tells you a little bit about us. We have about 4 to 6 minutes of ATP at any one time. Our cells don't store large quantities of ATP. So you've got a little bit in storage. You've got a few minutes worth in storage. But when you stop breathing, when you stop getting oxygen, you're on the clock towards death. And that's why we have to get air into your lungs as quickly as possible. All right. So with that little preamble, let's go into the lab slides and let's take a look at how microbes, particularly bacteria, use oxygen or don't use oxygen. So that's what you should be seeing now on your screen. You should be seeing the title slide for our lab today, which is called Oxygen and Growth. I'm just doing a little resizing here. Okay. So our objectives for today are first to identify the 5 different oxygen categories. In other words, the 5 kinds of relationships that microbes can have with oxygen. There are some microbes that are like us. They're called obligate aerobes. Those organisms have an absolute requirement for oxygen. So obligate, and aerobe refers to oxygen. Now, there are also obligate anaerobes. There are what are called aerotolerant anaerobes. There are microaerophiles. And finally, there are facultative anaerobes. We've briefly mentioned this last category in another lab. So we'll identify and we'll define these 5 categories. And then we're going to talk about a very special kind of medium called thioglycolate medium. And we'll look at how microbes grow in thioglycolate depending on which oxygen category they belong to. We'll talk about how we grow obligate anaerobes, what methods we have available to grow anaerobes. We'll talk a little bit about a diagnostic test called the Catalase test and how it relates to oxygen. And then we'll do a little experiment. We will take 4 different bacterial species and we'll inoculate them into thioglycolate. And we'll see how they grow and see if we can assign them to their oxygen category. So remember, all living cells require a constant source of ATP to use as energy currency. Remember, ATP stands for something. It's adenosine triphosphate. Different metabolic pathways can be used to make ATP. Absolutely. There's a process called aerobic cellular respiration. That's what you and I do. There is a process called anaerobic cellular respiration. Some microbes use that. And finally, fermentation is a metabolic pathway that will produce ATP. Now, we can test microbes in the microbiology lab to determine which oxygen category they belong in, what their oxygen requirements are. And we can also figure out which metabolic pathway they're using. We can adjust, in other words, we can adjust the conditions in culture according to the needs of the microbe. Up here at the top of the slide, this is our friend ATP, adenosine triphosphate. This molecule right here, this is a sugar. This is a 5-carbon sugar called ribose. This is a what we would call a base. This is the same kind of a base that you find in DNA and RNA. And then over here, we've got 1, 2, 3 phosphate groups. It's the phosphate groups that do all the work as ATP is built and then ATP is spent or broken down. It's the bonds, the covalent bonds between the phosphate groups where that little bit of energy is stored. And by breaking the bond between this outer phosphate group and the rest of the molecule, the cell can release a little bundle of energy to help it do work. All right, let's talk through these categories, these oxygen categories. The first group we said are the obligate aerobes. These are organisms that require oxygen to make their ATP. So again, you and I are obligate aerobes, and so are a lot of different microbes. They have an absolute requirement for oxygen. That's how they make their ATP. They use a process called aerobic cellular respiration to make ATP. Now, some obligate aerobes can also perform fermentation. It depends on the species that you're looking at. Can humans do fermentation? What do you think? Type it into chat if you know. Can you and I ferment? Can we do fermentation in our cells? We can do cellular respiration. That's the metabolic pathway we use most in order to make ATP for the needs of our cells. Do you know if we can ferment? Anybody have any idea? The answer is yes. Humans can ferment. Now, it's a secondary pathway for us. We don't use it to make our ATP. But if you get into trouble, if you cannot get enough oxygen, your cells will ferment. And the type of fermentation that human cells do is called lactic acid fermentation. We'll talk more about fermentation when we talk about microbial metabolism. But yeah, we can ferment. Now, lactic acid is the product of that fermentation process. And it builds up in your muscles and then in your blood when you're fermenting. Lactic acid is painful in your muscles. So imagine, I always use this example, imagine the person who comes out on a nice spring day, on a Saturday, and goes for a run. This person who has not gone running all winter, but a nice spring day comes and they put their shoes on and they go for a run. And you've probably seen these people on the side of the road running along, looking like they're dying, right? Gasping for breath as they run. That's a person who is asking their body to do a whole lot of work. So they need a lot of ATP, but they are unable to get enough oxygen to meet the requirement to make all that ATP. They're gasping because they cannot get enough air in for the amount of work they're doing. That person's cells are going to start to ferment, and they'll start to make lactic acid, and lactic acid hurts. That's part of the reason your muscles hurt when you push them like that, because you're fermenting. Now, as soon as that person stops and holds on to the telephone pole and leans over and gasps for air, they're gonna stop fermenting once they start getting enough oxygen again. But yeah, that's what we do when we get into trouble like that. When we're not getting enough oxygen, we can ferment. Now, fermentation, as we're going to learn, fermentation is a terrible way to make ATP. It's very inefficient. So that's why we only use it as a secondary pathway. Now, the other thing to note about obligate aerobes is this. We have the catalase enzyme available. You're going to note that any microbe or other organism that uses oxygen in any way is also going to have this little enzyme available in their cells, catalase. Catalase is an enzyme that helps get rid of a molecule called hydrogen peroxide. Oxygen is critical. Oxygen is life-saving in an obligate aerobe, but oxygen is also somewhat of a dangerous molecule in cells because it can create lots of different intermediate molecules that are toxic. And one of them is a molecule called hydrogen peroxide. So catalase helps our cells break down toxic hydrogen peroxide. Now, the second category is called an obligate anaerobe. Anaerobe meaning without oxygen. So these are organisms that cannot use oxygen to make ATP. And in fact, oxygen is toxic to them. These are organisms that use anaerobic cellular respiration and sometimes fermentation. It depends on the species. And these organisms don't have catalase. They don't need catalase because oxygen's toxic to them. That means oxygen is just never present in their cells. And they just don't need a way to get rid of dangerous oxygen intermediates like hydrogen peroxide. Now, I said a minute ago that cellular respiration involves breaking apart glucose, taking it all down to its most basic component. The cell breaks the glucose down bond by bond by bond. It breaks those covalent bonds and it releases the pair of electrons that are sitting in that bond. Remember, that's all a covalent bond is. It's a pair of electrons that are being shared by two atoms. So when a cell breaks down glucose, oxidizes it, that's the technical term, it breaks the covalent bonds holding that molecule together, and it releases the electrons. The cell is going to harvest the energy from those electrons and use it to make ATP. But it needs a place for those electrons to go. Some other molecule has to take those electrons, accept those electrons, get reduced. That's the term we use. If they're not using oxygen, what are they using? What are they using? The answer is anaerobic microbes can use one of several different molecules at the end of their electron transport chain. They use a molecule called nitrate. That's a very common one. Nitrate. There's another one called sulfate. There's one called carbonate. There are several different final electron acceptor molecules that anaerobes can use. So they're still doing cellular respiration, just like you and I are, but they're using a different molecule as the final electron acceptor. You and I use oxygen, we're aerobes. Anaerobes can use nitrate, sulfate, carbonate, and even other molecules, depending on the species. Now, the third category is called aerotolerant. So the name gives this one away a little bit. These are organisms that can't use oxygen to make their ATP, but they tolerate oxygen. An obligate anaerobe finds oxygen toxic, but an aerotolerant anaerobe will tolerate the oxygen. It's not going to use it, but it's okay to keep it around. It's not going to harm the microbe. Now, What's interesting about aerotolerance is this: they lack catalase. They don't have the catalase enzyme to break down hydrogen peroxide, but they don't have the whole system that makes hydrogen peroxide from oxygen. So hydrogen peroxide is not a problem for them. Remember, usually if a cell uses oxygen, if a cell has oxygen around in its environment, that cell has to be careful because one of the byproducts of oxygen is hydrogen peroxide. So usually when we see an organism that has oxygen around it in its environment, in its cytoplasm, we expect it to have catalase because the catalase will break down hydrogen peroxide. But this organism, this aerotolerant type of organism, it doesn't even make hydrogen peroxide. So it doesn't need an enzyme to break it down. Now, aerotolerant anaerobes typically use fermentation to make their ATP. And it turns out that these organisms grow best when there's a little bit of oxygen around, low oxygen concentration. What an interesting group of microbes. They grow best when there's a little bit of oxygen around, but they're not using the oxygen to make their ATP. Oxygen is not toxic to them. They tolerate it. And the metabolic process that they use to make their ATP is fermentation. Really interesting group of microbes. Next on the list, hold on just a sec. Oh, before I go to the next one, I said a minute ago that we can use a type of medium called thioglycolate. Thioglycolate is interesting because it is the first time we're going to see a semi-solid medium. It looks like a broth in these drawings. It looks like it's in test tubes, which it usually is, and it looks like a broth. But it's actually considered semi-solid because we put a little bit of agar in it. We put a tiny bit of agar into the broth just to make it a little bit more solid. It's not like agar, but it's not a pure liquid. It's just a little bit somewhere in between, semi-solid. And the reason thioglycolate is so useful to us is because of this thioglycolate chemical. Thioglycolate broth is going to have oxygen present in it at different amounts, depending on where you are in the tube. It's kind of neat. It allows oxygen to penetrate up at the top of the tube. So there's plenty of oxygen up at the top of the tube. But then as you start to go down deeper into the tube, there's less and less oxygen. Until you get down the bottom, there's very little oxygen down at the bottom. We use thioglycolate by inoculating it and then seeing where the microbes grow. Because they're going to grow at the level in the tube where they find the oxygen they need or don't need. They'll grow at certain levels in the tube depending on their oxygen requirements. Very, very useful media. So take a look at the ones we've talked about so far. Tube A is a drawing of the way that an obligate aerobe will grow inside thioglycolate. See how the little red balls, those represent cells. See how they're all up at the top of the tube? This is the place where oxygen is freely diffusing into the broth, into the medium. This is what we call the oxic zone. Oxic, O-X-I-C, oxic zone. This is where the most oxygen can be found. And an obligate aerobe must have oxygen to make its ATP. So it grows right there in the oxic zone. Now, if you look at the next one, this is how an obligate anaerobe grows, way down at the bottom. They're not going to grow up here because there's too much oxygen up here for them. They go way down to the bottom where there's very little oxygen, if any. I have a note down here too that cells often clump together when they're growing in thioglycolate. They're not evenly dispersed like they would be in a broth. They clump together and sometimes they grow as these sort of tendrils, these sort of snaky lines of cells. We'll see this in just a few minutes. Now, the other group that we looked at a moment ago were the aerotolerant anaerobes. Now, this drawing isn't as good. I had to modify it a little bit from what the artist had drawn. But remember, aerotolerants like a little bit of oxygen around. Not a lot, not what you would normally find in air, but they like a little bit. They grow best in a little bit of oxygen, even though they are not using the oxygen to make their ATP. So what you see when you grow an aerotolerant anaerobe in thioglycolate is this pattern. Most of the cells will be growing in the bottom half of the oxic zone. And you will also see some growth in the rest of the tube below it. So they can grow where there's no oxygen. They can grow where there's a little bit of oxygen. They can grow where there's a little more and a little more. They grow especially well in the bottom half of the oxic zone. That's what the red bar indicates. Now, the fourth category is called the Microaerophile. Now, if you translate this word, it gives you a lot of information. Remember, anytime you see the word phile or philic, that means loving. So a microaerophile is an organism that likes a little bit of oxygen. They need oxygen to make ATP, but at a reduced concentration. Okay, so remember, aerotolerant organisms, they like a little bit of oxygen around, but they don't use it. Microaerophiles do use it. They have to have it, but they need it at a reduced concentration. Microaerophiles grow best in a low oxygen concentration and also a high carbon dioxide concentration. Normally, there's about 5% carbon dioxide in the air. A microaerophile likes that between 5 and 10% carbon dioxide. So how do you grow an organism like this? Well, we have a sort of an old-fashioned tool called a carbon dioxide jar. And this just looks like a regular glass jar, but it has a packet of material in it. When you open up that packet of, when you close the jar, put the culture container into the jar and you close the jar, you tear open this packet before you close the jar. And what the packet does is it absorbs most of the carbon dioxide and most of the oxygen, but not all of it. So you end up with an environment that has the right amount of carbon dioxide, low levels of oxygen, and the organism grows great. In the old days, before we had the carbon dioxide generating packet in there, we used to put a candle in the jar. Now, it didn't help get the carbon dioxide levels right, but what a candle does is burn up the oxygen, right? Gets rid of the oxygen in the jar. The candle will glow and burn, and then it will go out when the oxygen levels are low enough to support the growth of those microbes. So it has a packet in it that will generate the extra carbon dioxide and absorb some of the oxygen, get some of the oxygen out of the air. Now, the fifth oxygen requirement group are called facultative anaerobes. And I have the asterisk here because most bacterial species belong to this group. Most of the bacteria are facultative anaerobes. And here's what their requirements are. They grow best when oxygen is around. They can use oxygen to make ATP, but they don't have to have oxygen to make ATP. They are facultative. They can switch back and forth between pathways depending upon what's available to them. They can do aerobic cellular respiration. They can do anaerobic cellular respiration. They can do fermentation. They can adjust to basically whatever environment you give them. If there's oxygen around, great, they'll use it. They'll do aerobic cellular respiration. If there's no oxygen available at that moment, well, they'll switch over and they'll do anaerobic cellular respiration, or maybe they'll do fermentation. It depends. It depends on the situation. So they're very adaptive. And as you can imagine, that helps them survive in a lot of different environments, a lot of different environmental conditions that they might encounter. Now, if we go back to our drawings here, this one in the at the very end is a microaerophile growing. This is what I mean when I say cells growing in the bottom half of the oxic zone. See how there's some broth, or I should say media. There's some media up here that doesn't have cells growing in it. But at the bottom of the oxic zone, that's where the growth is. So these cells are growing where there is a low concentration of oxygen. They can't grow through the rest of the tube because there's not enough oxygen. And they can't grow up top because there's too much oxygen. They're growing right at the sweet spot, right where there's enough oxygen for them, but not too much. Now, if you look over at the facultative anaerobe tube, you see this. Facultative anaerobes can grow throughout the entire tube because, like I said, they can do several different metabolic pathways. But notice that most of the growth is at the top. Facultative anaerobes will do aerobic respiration, aerobic cellular respiration, if there's oxygen available. So that's sort of their go-to metabolic pathway. If there's oxygen around, they'll use it. But if there's no oxygen around, they'll switch. They'll switch to anaerobic cellular respiration, or they'll switch to fermentation. That's why you see most of the growth at the top of the tube. So as you can see by looking at this drawing, if we have a microbe and we don't know what it is, and we don't know what its oxygen requirements are, if we put it into a thioglycolate broth, by looking at how it grows, we can figure out what oxygen category it belongs to. And we get another piece of information that will help us identify that microbe. Now, I want to show you how we do this Catalase test. Catalase testing is very rewarding because it's very easy and you get immediate results back. This is a test, remember, that looks for the presence of an enzyme in the microbial cells. And that enzyme is called catalase. Catalase breaks down hydrogen peroxide, that's H2O2. It breaks it down into water and oxygen gas. So the way you do this catalase test is you take a glass slide and you put just a loop full of culture material onto the slide, and then you apply a drop of hydrogen peroxide on top. Just regular old hydrogen peroxide from the drugstore. If that microbe, if those cells have catalase, what'll happen is it will immediately bubble up. It'll immediately start to foam up like this. The foam that you're seeing, the bubbles that you're seeing are oxygen gas. So this is a slide that has microbial cells on it that have catalase. They are breaking down the catalase. Sorry, they're breaking down the hydrogen peroxide with the catalase, making water and oxygen gas. And the oxygen gas is being liberated, and it causes the drop of fluid to foam up. Over here, this is what it would look like if the microbe did not have catalase. There was a loop full of cells here. We put a drop of hydrogen peroxide on top of them, and nothing happens. That's a negative. Very easy to read this test. You get immediate results. It's very rewarding. So here's a question for you. Here's a question from previous lecture material. You and I have catalase. We have oxygen in our cells all the time. So it makes sense that sometimes we have hydrogen peroxide developing and we need catalase to get rid of it. So where do we keep our catalase? Which organelle in human cells stores this catalase enzyme? Does anybody remember? The name of the organelle gives it away a little bit. Which organelle contains the catalase enzyme that helps get rid of hydrogen peroxide? Amy's got it. The peroxisome. Peroxisome. The name gives it away. Peroxisome sounds like hydrogen peroxide. And one of the things that is inside the peroxisome is the catalase enzyme. So that catalase enzyme helps our cells get rid of hydrogen peroxide if it forms. That was a good guess, Sop, the lysosome. But the peroxisome is really specialized. And the peroxisome is where this catalase enzyme is stored. Now, I mentioned at the start of lab that one of the things we have to be able to do is grow anaerobes in the lab. And as you can imagine, this is a little bit difficult because these obligate anaerobes especially have trouble growing because they find oxygen toxic. Now, we have to exclude oxygen from their environment in order to allow them to grow. And there's a couple of ways we can do that. One of the most common ways is to use a tool called a Brewer's jar or a Brewer's anaerobic jar. And I'll show you a picture of one of these in a minute. These are also glass jars or plastic jars that have a tight fitting lid. And they also come with this little pack of material. But this pack contains hydrogen gas packs or hydrogen gas generators. So you tear open this little packet, put it in the jar, and then you put your culture container in, close the lid very tightly, and the packet is going to generate hydrogen gas. What'll happen is the hydrogen will combine with any oxygen that's present in the jar, and it will make water. So within a few minutes, you've got no oxygen in that jar anymore. Now, the other thing we often add into the Brewer's jar is a second kind of packet of material, which is called an oxygen indicator packet. The second packet sits in the jar while the incubation period is occurring. And if any oxygen accidentally gets into that jar, let's say the lid wasn't put on quite tightly enough. If any oxygen gets in during that 24-hour incubation, the oxygen indicator will turn color. It will change colors. It usually changes to a pink color. That'll tell you that, uh-oh, something went wrong. Oxygen got into the jar, and that's probably why you're not going to get any growth. So you get 2 packets in a brewer's jar. One packet generates hydrogen gas, and the hydrogen gas will react with the oxygen in the jar and make water. The second packet is an oxygen indicator, and if any oxygen accidentally gets into the jar, it'll turn pink. So brewer's jars are very helpful. They're low tech, they're not expensive, very, very helpful to us in the lab. Now remember, technically speaking, we can also use thioglycolate broth to grow anaerobes. It's not an ideal way to do it, but you can grow anaerobes in thioglycolate like we just saw. And that's because of this compound called thioglycolate. Remember, this has a small amount of agar in it. And what that agar does is help prevent oxygen from penetrating all the way to the bottom of the tube. You will get an oxic zone at the top where there's lots of oxygen, but then there'll be less and less as you go down until there's none at the bottom. Sodium thioglycolate is a reducing agent. It's a reducing agent. It accepts oxygen and it takes oxygen out of the deeper regions of the broth tube. Now, one other thing to know is you can buy thioglycolate broth from a chemical supply company. And sometimes it's helpful to ask them to put an indicator dye into it. Remember what indicator dyes do. They change color depending on the pH. When you put an indicator dye into thioglycolate, it will show you exactly where the oxic zone is. In other words, when you have a thioglycolate tube that has indicator dye in it, up at the top of the tube in that oxic zone, the medium will be pink. So you'll know exactly where the oxic zone is. And then when you put your cells in and you grow your cells, the next day you'll know for sure if they're in the oxic zone or they're out of the oxic zone, or maybe they're in the bottom half of the oxic zone. It just gives you a much clearer indication of exactly where the oxic zone is. One other thing to note is that when we're working with anaerobes, there are anaerobic incubators available to us. And there are also what we call glove boxes available to us. That's what you're looking at in this picture over here. This is just a chamber that we can adjust the amount of oxygen in. And you can see that there are gloves here so we can reach into the chamber and manipulate our culture containers without opening the door up. So these glove boxes can be used to generate either a low oxygen or a no oxygen environment. And you can also buy incubators that have the ability to generate no oxygen environments. As you can imagine, these anaerobic incubators, they're very expensive. These glove boxes aren't nearly as expensive. But if you worked in a lab that worked with a lot of anaerobes, it would be absolutely worthwhile for them to invest in an anaerobic incubator. On this slide, up at the top on the left, this is what a brewer's jar looks like. So you can see it's just a glass or plastic jar, and it's got a lid on it that also has a handle with a thumb screw. So when you put the lid on, after you screw the lid down, you turn this thumb screw down to make sure that the lid is as tightly attached as possible. Over here on the right, this shows the packets of material that come with this jar. One of these is the hydrogen gas packet, and one of them is the indicator, oxygen indicator packet. Notice too that the diameter of a brewer's jar is just wide enough to fit a Petri dish. So what people usually do is they stack the Petri dishes up in here. They're going to fit right in that jar, right on top of each other. This is what thioglycolate broth looks like before you inoculate it. It looks just like a lot of our other broths. And over here, this is thioglycolate broth that has an indicator dye in it. So you can clearly see where the oxic zone is, where there's a lot of oxygen present, plenty of oxygen present. The oxygen levels get lower and lower and lower as you go deeper into the tube. And then you get to the anaerobic zone, no oxygen. Does anybody have any questions so far about these 5 oxygen categories? We have obligate aerobe, obligate anaerobe, aerotolerant, microaerophile, and facultative anaerobe. All right, very good. Before we take a look at our little virtual experiment today, we'll take a short break. Okay, I'll see you back here at 10 o'clock. Okay, we'll see you in a few minutes. Hi, everybody. Welcome back. Go ahead and type your name into chat for me. Thank you. Thank you very much. Thank you. That's great. All right. All right. What we're going to do now is have our next virtual experiment. We're going to do an experiment with some certain species of bacteria, and we'll use some thioglycolate broth to see what we get for results. So for today's exercise, we're going to use 4 different organisms. Some of these you've seen before now, but there are a couple new ones today. The first organism is called Clostridium sporogenes. This is a gram-positive rod, and this is an organism that can live in the human body as well as out in the environment. So you'll find Clostridium sporogenes inside the gastrointestinal tract of mammals like us, but you can also find it out in the soil, and you can find it in certain sediments like in the ocean. You can find it in the sediment at the bottom of the ocean. Here's just a nice image, a stained smear of Clostridium sporogenes. So you can see that these are rod-shaped organisms. You can definitely see that rod shape. It's hard to say that they're doing any particular kind of arrangement here. This is an organism that's not known to have any particular type of arrangement in culture. Next on the list is an organism we saw last time, which is Enterococcus faecalis. Remember, this is a gram-positive coccus, and you'll notice that I wrote coccobacillus here as well. Some scientists have reported that there are strains of Enterococcus that actually don't look like an ordinary coccus. They're not spherical so much as they are more egg-shaped, more like a coccobacillus. In my experience, they're usually cocci. These are organisms that, again, live in the mammalian gastrointestinal tract. You can also find them, though, in the mouth. You can find them even in the vagina. These are commonly found in the human body. Here is this organism under the microscope. I want you to notice how, if you look around here for one cell, it's kind of hard, right? The smallest groups that you tend to see are like this, 2 cells side by side. Here's another one, 2 cells side by side, 2 cells here. You can see where somebody might look at this and call this a coccobacillus. It's a little bit more oval or egg-shaped than other types of cocci, but definitely a gram-positive organism, and it can be found in the GI tract. Here's our friend E. coli. Remember, E. coli is a gram-negative rod, also found in the gastrointestinal tract. E. coli under the 40x lens is kind of hard to see. These are small, stubby little rods. Up here in the corner, they've shown us underneath the 100x lens, so you can see the rod shape. And the last organism we're using today is called Alcaligenes. Alcaligenes faecalis. So another fecal organism, but like Clostridium, it can be found out in the environment as well. You can find Alcaligenes in the soil, in certain bodies of water, but it is a gram-negative rod. And when you look at Alcaligenes underneath the microscope, you can clearly see it's gram-negative. It's a little harder to see the shape in this particular smear because we're only under the 40x lens here. So 4 organisms that all can be found in the gastrointestinal tract. 2 of them are gram-positive and 2 of them are gram-negative. I'll let you know that all 4 cultures were confirmed to be alive. So we took some of each one and streaked it out on a nutrient auger. You should also remember that just because something lives or can live inside the gastrointestinal tract, some strains, some species and strains are non-pathogenic, but some can be pathogenic. Some of them can cause disease. Now, for the media we're using today, we're going to use thioglycolate broth. So we'll inoculate some thioglycolate tubes. We're also going to use plain old nutrient agar. Remember, that's a basal medium. And we'll put the microbes onto nutrient agar so that we can compare their growth in room air versus in a brewer's jar. Remember, room air is about 21% oxygen. Whether you're in the incubator or out of the incubator, you've got about 21% oxygen in regular air. In the brewer's jar, we're going to make sure there's no oxygen. And finally, we'll also perform some catalase tests. All right. The first thing we need to do is think through our 4 organisms and make some predictions. So first thing to think about, what can grow or what should be able to grow on nutrient agar? That's what the Na is. If we incubate in regular old room air, which of our 5 categories, our 5 oxygen categories, would we expect to grow on a nutrient agar plate that was in room air? Well, we know the obligate aerobes will grow, right? Because they love oxygen. They need oxygen. They have to have oxygen or they won't survive. So certainly the obligate aerobes. Now, the other group will be facultative anaerobes because we said they can live in virtually any environment. What about aerotolerant and microaerophile? Remember, these are 2 groups that prefer to be in low concentrations of oxygen. The aerotolerant group, it doesn't use oxygen. It doesn't find oxygen toxic, but it prefers to be in a low oxygen environment. Microaerophiles require oxygen, but again, they need it at a low concentration. It turns out that both of these categories of organism, they will grow in air, but they grow poorly. They're going to grow poorly when they're exposed to regular air. And that's because there's just too much oxygen in regular air for them. Remember, when we say something grows poorly, what we're saying is on a plate, the colonies would be really small. And we wouldn't see as many colonies as we might expect. And in a broth tube, it just wouldn't be as cloudy as we might expect it to be. We're just not going to get normal amounts of growth. That's poor growth. So in room air, there's only one oxygen category missing, and that's the obligate anaerobes. The only group that can't grow in room air is the obligate anaerobes. So the next question is, what would we expect to be able to grow on a nutrient agar plate in a brewer's jar where there is no oxygen? Well, that's where the obligate anaerobes will grow because they're the ones that cannot be around oxygen. Oxygen's toxic to them. So we certainly expect them to grow in a brewer's jar, but they're not the only ones. Remember, facultative anaerobes, they kind of adjust to their environment. So a facultative anaerobe can grow in a brewer's jar because they can use anaerobic cellular respiration if they have to. They can also ferment if they have to. There's one more group that'll grow in a brewer's jar, and that's the aerotolerants. Aerotolerant anaerobes, remember, they don't mind being around oxygen. They just don't use it. They don't need oxygen to make ATP. They're not going to use the oxygen, but they can be around it. Remember, though, they're going to grow poorly in a brewer's jar. They're going to grow poorly because they prefer to be in low oxygen environments. They grow best in low oxygen environments. And in a brewer's jar, there's no oxygen. Now, let's also think about the catalase test. Which groups should have a positive catalase test? Obligate aerobes, of course, because they're around oxygen all the time. Facultative aerobes will also be catalase positive, as will the microaerophiles. These are groups that we would expect these cells to have oxygen around them. Which groups should have a negative catalase test? The obligate anaerobes and the aerotolerant. Remember, the aerotolerant group is a little bit special because they don't have the ability to even make hydrogen peroxide. They can't do the chemical reaction with oxygen that makes hydrogen peroxide. So there's no need for them to have catalase around because there's no hydrogen peroxide to break down. Obligate anaerobes, they're also going to be catalase negative because they can't be around oxygen. We would never expect there to be oxygen floating around in their cytoplasm. Remember, we can look at our thioglycolate tubes and we can know how these microbes are relating to oxygen. Obligate aerobes are going to be all the way through the oxic zone. Obligate anaerobes are going to be at the very bottom of the tube. Facultative anaerobes are going to be all the way through the tube, but most growth is in the oxic zone. Aerotolerants will grow all the way through the tube, but only in the lower half of the oxic zone. And microaerophiles will only grow in the lower half of the oxic zone. All right. I put each one of our microbes onto a nutrient agar plate, and I grew those, or I attempted to grow those nutrient agar plates in room air and also in a brewer's jar so we can compare. And here are the results. The Clostridium sporogenes microbe, I got no growth in room air. I got growth with good-sized normal colonies in the brewer's jar. No growth in room air, not a single colony grew. But in the brewer's jar, I got plenty of colonies. Lots of colonies I would call normal growth. The colonies were normal sized. There was a good number of them, what I would consider normal growth. Any ideas which group Clostridium sporogenes might belong to? Type it right into chat for me. They cannot grow in room air. I didn't even get pore growth. I didn't even get pore growth in room air. So ask yourself, are there any groups that cannot grow in room air? Remember, we said 4 groups can grow in room air. 1 group can't. Only 1. Sarah's got it. Candy's got it. Obligate anaerobe. The obligate anaerobe is the only group that cannot grow in room air. Obligate aerobes will grow great in room air. Facultative anaerobes, they'll grow great in room air. Aerotolerants, they'll grow, but they grow poorly. Microaerophiles, they'll grow, but they grow poorly. But obligate anaerobes, they cannot grow in room air because oxygen is toxic to them. Enterococcus faecalis, in room air, it grew, but it grew poorly. In the brewer's jar, it grew great. It grew with good-sized normal colonies. What do you think? It can grow in room air. It can grow in the brewer's jar. In room air, let me pull it back up for you. In room air, it doesn't grow well. It grows poorly, small colonies. In the brewer's jar, it grew great. Any ideas? There's only two groups that will grow in room air poorly. Aerotolerant, microaerophiles. They grow poorly in room air. What about in the brewer's jar? What groups grow great in the brewer's jar? We've got a vote for facultative anaerobes. We have a vote for aerotolerant. Any votes for microaerophiles? We know that both aerotolerants and microaerophiles grow poorly in room air. So those should be your top thoughts, that it's either an aerotolerant or a microaerophile. Remember, Amy, facultative anaerobes grow great in room air. They grow great. Let's take a look. What can grow in a brewer's jar? Obligate anaerobes, of course, facultative anaerobes, and aerotolerants. So we have two possibilities that grow poorly in room air, and only one of them can grow in the brewer's jar. We don't really know yet, do we? We're kind of on the edge. It looks like aerotolerant, but aerotolerants, remember, when we put aerotolerants in a brewer's jar, we expect them to grow poorly. We expect them to grow poorly in room air. What could this be? Might be aerotolerant. We'll have to wait. E. coli. E. coli grew great in room air. E. coli grew in the brewer's jar, but not great, poorly. Alkyligenes faecalis grew great in room air, did not grow in the brewer's jar. Think about the alkyligenes for a minute. Grew great in room air, didn't grow at all in the brewer's jar. Which group cannot grow without oxygen? Which group cannot grow without oxygen? We got no growth in the brewer's jar. Which group grows great in air, but cannot grow in a brewer's jar? Obligate aerobes. Good job, Candy. Yes. So alkyligenes seems to be clear. Clostridium seems to be clear. Those are our obligates. But let's see what other data we can gather about the other two. This slide is just a repeat of what I showed you a moment ago about what can grow in air, what can grow in a brewer's jar, what we would expect. Take a look at the thioglycolate results. Now, one thing I want you to remember is that this is a semi-solid medium. And sometimes when the cells grow, they start to form these tendrils. They're not evenly distributed, in other words, throughout the medium. They tend to clump together and make these long strands of cells that are growing clumped together. Here's Clostridium sporogenes. Now, it's hard to tell in the picture, but I'll point it out to you. This is my thioglycolate tube. This right here is a little clump of cells growing at the bottom. A little white clump of cells growing at the bottom. Remember, we said Clostridium sporogenes cannot grow in room air, but it grows great in a brewer's jar, and it grows at the very bottom of my thioglycolate tube. Not very well, I might add. It's kind of struggling. What do we think Clostridium sporogenes is now? It does not grow in room air. It grows great in a brewer's jar, and it grows at the very bottom of the thioglycolate tube in a place where there is no oxygen. Which group would you put Clostridium sporogenes in? Grows great in a brewer's jar. Does not grow at all in room air, and grows at the very bottom of the thioglycolate tube. There's only one group that this organism fits neatly into. Remember, Sarah, we can't just say anaerobe. We have to put a qualifier with anaerobe. There are obligate anaerobes. There are facultative anaerobes. And there are aerotolerant anaerobes. Austin's got it. Candy's got it. This organism is an obligate anaerobe. It does not use oxygen to do its ATP production, and it finds oxygen toxic. It will not grow when it's exposed to oxygen. It likes an environment where there is no oxygen. So that's brewer's jar and the very bottom of the thioglycolate tube. Here's Enterococcus, and you can really see these tendrils in this particular tube. See how you've got sort of a mat of growth here? This looks very different from a broth tube. Remember, this is not pure broth. This has a little agar in it. So it kind of forces the cells to clump together when they grow. We got this big, big mat of growth right here. But we also have growth down through the rest of the tube. There's just less of it. So this organism is not growing at the very top. It's growing in the bottom half of the oxic zone. In fact, it's growing very well in the bottom half of the oxic zone. That's where most of the growth is. But I also have growth reaching down into the rest of the tube. What do you think about this one? Does it meet any of our drawings? What do you think? We have growth. We have a big, heavy mat, a big clump of growth in the lower half of the oxic zone. Not up top, though. It's not growing all the way through the oxic zone. So it clearly has a special requirement for oxygen levels. But it also is growing down into the rest of the tube. So it can grow in low oxygen and it can grow in no oxygen. What do you think? Candy's thinking maybe facultative anaerobe. Sop is thinking maybe aerotolerant anaerobe. Let's compare those two. Here's the facultative. Here's the aerotolerant. Remember, I've crudely drawn on here what is supposed to represent no growth at the top of the oxic zone and heavy growth in the lower half of the oxic zone, and then growth through the rest of the tube. That's aerotolerant. The facultative anaerobes will grow throughout the oxic zone. In fact, they'll grow best throughout the oxic zone, and they'll also grow through the other regions of the tube. What do you think? Did we get any growth at the very top of that tube? Let's look again. Look at this tube. We very clearly have a region at the top of the oxic zone where there's nothing growing. This is the growth, this yellowish, whitish material that you see in this tube. These are the cells growing and clumping together. And then you see these sort of tendrils hanging down into the deeper reaches of the tube. This long tendril goes all the way down to the bottom. These are cells, they're all clumped together, they're stuck together in this semi-solid medium. We have most growth right here in the lower half of the oxic zone, but we also have some growth down through the rest of the tube. This is the giveaway right here. There's nothing growing at the very top of the oxic zone. That's the giveaway. That tells you that this can't be a facultative anaerobe. A facultative anaerobe would grow all the way through the oxic zone. Okay? This is an aerotolerant anaerobe. This is an organism that doesn't need oxygen to grow, but it tolerates oxygen. And it grows best in low oxygen environments. All right. Let's see what else we get in our thioglycolate tubes. I have two E. coli tubes to show you because neither one came out particularly well. And also I have our alkylagenase faecalis tube. Let's take a look at E. coli first. Here's one E. coli tube. Look how strange the growth pattern looks. Those tendrils are so strange, aren't they? Here's another one. So what you can see in this tube over here, I can see this whitish material up at the top of the tube, but it's hard to tell if it goes to the very top. There might be just a little bit of liquid up here that doesn't have any cells growing in it. It's hard to tell, but I've also got tendrils reaching down into the lower parts of the tube. Not all the way to the bottom though, it looks like, just kind of halfway. Very strange looking tube, this one. Compare it to this one, which also has E. coli growing in it. Here you've clearly got a big amount of this growth, this whitish material in the oxic zone. In fact, all the way through the oxic zone. Now it's very hard to see, I know, but there is a tendril growing right here. And it goes all the way down to the bottom. So these two tubes, these are important. They show you something. Thioglycolate tubes, they can be a little hard to interpret, and it's because they're semi-solid. Sometimes you'll get a result that doesn't really follow the textbook. Sometimes you'll get a result and you'll be hard pressed to interpret it. in that first tube, does it grow all the way through the oxic zone? It's kind of hard to tell. It looks like there's a little tiny bit at the top where there's no growth. But that would mean that you've got heavy growth through most of the oxic zone. When I put the exact same organism in a second tube, it grew all the way through the oxic zone. So we don't usually inoculate thioglycolate broth into one tube. We usually do 2 or 3 tubes, just so we got so that we get something we can compare among the 3 and come to a consensus. So let's take a look again at E. coli and see if we can come to a consensus. In terms of the oxic zone, it's either growing all the way through the oxic zone, right? Or through most of the oxic zone. But I also have tendrils coming down into the lower reaches of the tube, at least into the middle areas of the tube here. What do you think E. coli might be? It looks different from the aerotolerant tube, doesn't it? The aerotolerant tube very clearly had growth in the bottom half of the oxic zone, and the top half had no growth. This one is more like all the way through the oxic zone and down into the rest of the tube. Candy's got it. Candy, we finally got to the facultative anaerobe. That's right. Yeah, this is, E. coli is a facultative anaerobe. E. coli prefers to grow in oxygen. If oxygen's around, it's going to use oxygen to make its ATP. But it's facultative. It can adapt. And that means that if there's no oxygen around, it can switch over and it can do anaerobic cellular respiration, or it can ferment. It has the ability to switch back and forth between those metabolic pathways in terms of making ATP. So it's a very, very adaptive organism. All right, the last one on our list was alkaligenes, Alkaligenes faecalis. And here's alkaligenes. Again, a little bit hard to see this tube. What do you think? I've got growth up here. I have no growth down here. Now, again, you might be able to think, oh, it looks like there's a tiny strip right up here where there's no growth. But I can tell you that this organism grew all the way through the oxic zone. But it doesn't have any tendrils hanging down. It grows all the way through the oxic zone, but it can't grow through the rest of the tube. Candy's got it. Amy's got it. Good job. This is an obligate aerobe. It's got to have oxygen. It must have oxygen to make its ATP. It will grow all the way through the oxic zone because there's plenty of oxygen in there, but it won't grow anywhere else because there's not enough oxygen. Remember, in the middle of the thioglycolate tube, there is some oxygen. It's low levels of oxygen. And at the bottom of the tube, there's no oxygen. So this is an organism that it has to have at least, a decent level of oxygen. And it prefers to be all the way through that oxic zone. It has to have oxygen. It cannot make its ATP without oxygen. Obligate aerobe. Good job. So by inoculating thioglycolate broth and looking at the pattern of growth, we can often get information about the organism's oxygen requirement category. Sometimes the results are very obvious. like when you see growth only in the oxic zone, or you see growth only in the bottom of the tube. We didn't have a chance today to see a microaerophile growing, but that one is also kind of easy to see. It grows in the lower half of the oxic zone, and that's the only place it grows in the tube. It can be a little bit confusing when you're looking at aerotolerant organisms or facultative organisms. The key thing to focus on is whether or not the top of the oxic zone has cells growing in it, because that's really the difference between these two organisms. So Clostridium sporogenes, no growth in the room air, but grew great in a brewer's jar and grew in the deep anoxic region of the thioglycolate tube. This is our obligate anaerobe. On the opposite end, we had Alcaligenes faecalis, which did grow in room air, but not in the brewer's jar, and grew only in the oxic zone. Here's our obligate aerobe. Enterococcus faecalis grew in room air, grew in the brewer's jar, but poorly. This is an organism that can tolerate oxygen, but doesn't need it to grow. This is an aerotolerant anaerobe. E. coli grew in room air, grew in the brewer's jar, but not as well. Grew all the way throughout the thioglycolate tube, but best in the oxic zone. This is a facultative anaerobe. Remember, most bacteria are facultative anaerobes. Any questions? This is one of the pieces of information that we can gather in the laboratory about a microbe that we're trying to identify. Just one piece. We have to do many tests in the laboratory if we want to identify the genus and the species. You can't do just one. Gram staining is very helpful. Gram staining puts that organism into one of two big categories, either gram positive or gram negative. It's very helpful to us, but we can't stop there. We have to do more tests. We have to use our special media like we did last time. We can use our thioglycolate broth. We can use our brewer's jar. We can test to see what kind of metabolic traits this microbe has. And that information gives us little bits and pieces about the microbe that when they're all added up, we can make a final decision about what the identity of that microbe is. It's also helpful for us to be able to open up a textbook and see, for example, that Clostridium sporogenes is an obligate anaerobe. Let's say we get a sample into the lab and we're told this is Clostridium sporogenes. We know that we're going to have to be very careful when we grow it, because that's an organism that cannot tolerate any oxygen around it. So we're going to have to set up the brewer's jar if we want to purposefully grow this organism. Depending on what your goal is, if you're trying to identify a microbe, or perhaps you're just trying to grow a microbe because you're studying it. You're in a research lab and you're trying to answer some questions about it. You have to know what its traits are so that you can grow it effectively. You know what to feed it and you know how to take care of it. It's a strange way to think about microbes, but that's part of what we do in the lab. All right, everybody, just a reminder, your lecture exam opens up tomorrow morning at 8 o'clock. Don't forget, you need to have that into me by Saturday at midnight. You also have some lab homework to do for me, which you should be able to do now without any problems. And don't forget your lecture quiz. That's also due tonight by midnight. Now, on Monday next week, we have a holiday, July 4th. So I won't be seeing you on Monday, but I will see you again on Wednesday. Don't forget to message me through Canvas if you have any questions or problems before then. Okay? And if there are no other questions, I'm going to let you go. Enjoy the rest of the day, everybody. We'll see you soon.